University of Gothenburg with Antonelli as my supervisor. So I will discuss some phylogenomics and evolution in this beautiful group in Asteraceae, the subfamily Barnadesioideae. But first of all, who is Barnadesioideae? Who are the Barnadesioideae? And how can we recognize this group? So, Barnadesioideae is a monophyletic subfamily comprising 90 genera and 84 species endemic to South America. So the species can be found from Venezuela into Argentina, and most of the species are found in xeromorphic areas along to the Andes and the Patagonia. Next. So the plants are very useful. I think everybody knows that Chuquiraga was chosen as the plant symbol of the present Latin American Congress. And if you go to some marketplace in Ecuador and especially in Peru, you can buy the leaves and the flowers, and you can buy your own tea. And with this tea, you can use it for many health treatments. So how can we recognize this group? We have two morphological synapomorphisms for this group. The first one is the presence of Barnadesioideae trichomes. So it's composed by 3 unbranched cells, and these trichomes are found on the corollas and the sip cells and the pappus. And another diagnostic feature is the presence of axillary spines. And because these spines are very hard, and the people usually call the plants as "don't touch me." And the spines can be found in pairs or in fascicles. Yeah. So, why do you study the "don't touch me" plants? Next one. I have many reasons to study this group, and I could spend one day just talking about that, but I will give some of those. First of all, it has a great diversity. The plants can be from herbs, subshrubs, shrubs, up to large trees up to 30 meters high, or also wood vines. And can be found from the sea level to the summits of about 4,000 meters high, in a great diversity of environments. So we can find the plants in some humid areas, like in the Atlantic forest, where most of the species are found in xeromorphic areas, such as in deserts, cerrado, and pampas and páramos. And many of the morphological features are adapted to the extreme conditions, for example, the leaves in Chuquiraga. So the second reason why I study this group is the phylogenetic position into the Asteraceae phylogeny. So in 1987, Jensen Palmer showed that all the Asteraceae have an inversion of 22 kilobase in the largest single copy, except this group that I studied. And shortly after, some phylogeny showed that my group, Barnadesioideae, is recovered as the sister group to the rest of the family. So if you investigate the morphology, the historical biogeography, and the morphology, we can provide insights into the early evolution of the family. But in the first year of my PhD, I downloaded many sequences from GenBank, and I tried to propose a phylogenetic hypothesis. But the branches in red, and you can see, it's my group. So I didn't have low support and resolution for the plates. So that couldn't answer my questions. And I decided to call to my supervisor in Sweden, so Alexandre Antonelli, and say, Antonelli, what can we do? No, I don't have resolution. I cannot answer anything. And he said, do you know that Mendel in 2014 developed probes for the family, and we can use that for your group? And I say, no, I'm not a bioinformatician. I cannot analyze the data. And he said, no, I have a PhD student develop a very user-friendly pipeline to analyze the data, and he can help you with your data if you come to Sweden. So that's it, sometimes Sweden. Okay. But when I started my PhD, this project, I was concerned with one statement in 2014. The most basal taxon, Fucaldaestucci, which had the lowest number of blast filter reeds, also had the lowest number of valvular contigs and cold blast reeds. even lower than the out group. So I could have many problems. Maybe the probes could not work for the group, and I spend my money and my time analyze the data, and who knows. So we decided to investigate many aspects of the phylogenomics. And the first one is, what is the impact of missing data on phylogenetic reconstruction? What is the difference? And because this group is mainly distributed in the Andes, so I'm a student, I didn't have money for collecting every South American countries, so we use many herbarium samples. So, but what is the difference between the herbarium and silica gel materials on phylogenetic analysis? And how are the genera of Barnadesioideae related? And are the genera of Barnadesioideae monophyletic taxa? So, to understand this, we sequenced 61 taxa in Barnadesioideae, comprising 61 taxa in your phylogeny, comprising 49 taxa in Barnadesioideae, and include 8 of the 90 genera and 45 of the 84 species in this group, and 6 subspecies. And we used some 11 species from Asteraceae used in the other articles, and 1 Caliceraceae species. And we have some herbarium samples and some silica gel samples, and we use the sequence capture processor to analyze the nuclear data. And we, as Mendel said, we didn't enrich our samples with using chloroplast data, but we were able to capture those data using mapping our sequence against to Centaura and Lactuca. So to decide to investigate the impact of missing data, we created 3 matrices of taxonomic completeness, but you can say I don't work with phylogenomics. I don't know what that means, so I can briefly explain. So imagine here you have a folder comprising 6 loci. So in the first approach, you can say to the phylogenomics, the program that I use it, combining all the loci comprising 3 or more taxa. So in this case, we have 6 loci. In the second approach, you can say combining only the loci greater than 50% of the taxa. So in this case, we have 4 loci. And the third approach, you can say to phylogenetic, combining only the loci comprising more than 75% of the taxa. So in this case, we have 2 loci. And the 3 matrices of taxonomic completeness, we inferred our phylogenies using concatenation and coalescent analysis. Okay, so let's see the results, the most important part. So across 3 or more loci, we were able to recover 967 loci. So it's a amount, right? It's a huge data set. But most of that matrix was a constant size. And we have a large amount of missing data, 86%. So as the matrix decreases in size, we can see that the number of constant sites and the amount of missing data decrease. And the number of variable sites and informative sites increase. So maybe we can think that this third matrix is the best to infer our phylogeny. I mean, most robust, more support, right? No. So here we have the average of the bootstraps. So as you can see, when you have the highest number of missing data, we also have the highest number of support nodes in both coalescent and coalescent analysis. So let's see these same results in another graph. So here we have the number of nodes across 4 different bootstrap thresholds. So we also have the 3 matrices of completeness in both concatenation and coalescent approach. So as we can see here, the highest number of support nodes is the matrix that comprises the highest number of missing data. And surprising, when you reduce your matrix, we can see that it received the lowest number of the high support nodes. And surprisingly, when you can see the unsupported nodes, we have the lowest number when you have the highest number of missing data. So I'm discussing this because when you use some pipelines, they say you need to cut your data because you don't like a matrix with a huge amount of missing data, but it's not true. So test your data set that you have in your head. So what about the herbarium and silica gel? So here we have 4 different parameters that we use. The number of raw reads, how much data we exclude because they are not in good quality, the number of loci, and the number of chloroplastins that we recover. So here we can see in general that we recover less data for the herbarium than the materials in silica gel. But is that statistically significant? Yeah, it is for both data sets, for 2 data sets, for the number of raw reads and nuclear loci. So here I'm saying that if you are thinking into investigate using nuclear loci, maybe herbarium is not the best option. But surprisingly, the quality of the herbarium samples were not so different when you compare to the silica gel. And if you are thinking using phylogenomics only for the chloroplast data, it's okay. So let's see the phylogeny. So across the 3 matrices of taxonomic completeness in both coalescence analysis and concatenation, it was highly congruent in measured clades. So in all phylogenetics, Barnadesioideae is recovered as a monophyletic group and sister group to the rest of composites. So another congruent clade was Barnadesioideae and Huarpia. Both genera are well defined. And this clade is supported by the basal of the anther is very short and by the lophate pollen. Another congruent result was that Dasyphilum is a clearly monophyletic group. So both species, 2 species of Dasyphilum subgenus Archidasyphilum are distributed in Chile and some areas of Argentina and have this apical appendage obitus and pinnate venation. The photo is not great, but and Dasyphilum sensu stricto has the bilobated apical appendage and the chordus venaceus. Actually, Dasyphilum was the subject of my master, and I sent the paper last month, so I have the combinations as soon as possible. So another congruent clade was that to both species that are put in another genus is always recovered as the sister group as Foucauldeia and Arnaldua. But this clade has a lot of morphology very different, and synapomorphism to this clade is still unclear. And another interesting clade that we cover is Chuquiraga and Donnerphyton. So in all the species tree, Chuquiraga is recovered as the sister group to Donnerphyton. But in the analysis using RexML, maximum likelihood, comprising all the loci, Chuquiraga is paraphyletic by the position of Donnerphyton as the sister group to section Chuquiraga. But this relationship receives a low support. And in the chloroplast data, Donnerphyton is recovered as the sister group to section Acanthophyllum. And this clade is supported by the smooth pollen and by the long caudate anthers. And another, this is the last one, I promise. And the last group that we would like to talk is the Schlechtsteindalia. The analysis comprising the lowest number of missing data, Schlechtsteindalia is recovered as the sister group to Donnerphyton in Chuquiraga, but these receive a low support. And in the chloroplast data, Schlechtsteindalia is recovered to the sister group to the large plate. So that's it. I'm still working because I'm a PhD student, and I hope not to break my arm because I hope you have some months, and thanks.